I will be talking about combating harmful algal blooms. So we are going to talk about what are harmful algal blooms? Do they occur in Maryland? What causes them? And what can we do to combat them? And what our lab is specifically doing to combat harmful algal blooms. In case anyone's curious, the Avengers do actually fight harmful algal blooms, just maybe not in the movies. This is a screenshot from the PlayStation 4 Spider-Man game, and he actually has to stop a bloom from occurring at the Hudson River. So he says, the algae is already killing fish, and if I don't stop it, it'll work its way up the food chain fast. I whipped up a virus that'll attack it in Harry's lab, and it's tailored to the toxic algae, so it won't hurt normal plant life, which is good because a park without plants is not a park. So this is just pretty much a summary of what we're going to be talking about today. Just, I wish we had Spider-Man who could focus in on and have Harry's lab whip us up a virus. So in order to talk about harmful algal blooms, I have to talk about phytoplankton and introduce phytoplankton to you, which is the algae we will be discussing today. Phytoplankton literally means drifting plants and can be eukaryotes or prokaryotes. And they are a base of the food web. So everything you see in the they occur in all water bodies, saltwater and freshwater. And yeah, so what are harmful algal blooms? Well, harmful algal blooms are any time water use is impaired. This could be either from the production of toxins, which are harmful to humans and other organisms, or it can just be when they reach such high densities that they cause low light penetration or low, and when they die, they cause low oxygen zones. This leads to fish kills and other harmful effects to the ecosystem. For instance, this is a picture of a fish kill that occurred this year in Spa Creek, Annapolis, where 10,000 fish died. So, although phytoplankton are very important to our food web, to the marine environment, there are these special class of phytoplankton that are harmful. So, as Spider-Man said, the algae is already killing fish, and if we don't stop it, it'll work its way up the food chain fast. So some toxins actually can biomagnify, meaning each small algal cell has very little toxin, but once those get eaten by zooplankton, and a bunch of zooplankton get eaten by fish, and then we eat the fish, we're exposed to a lot of the toxin. So this brings me to, why do we care about harmful algal blooms? Harmful algal blooms cause ecological impacts, economic impacts, and health impacts. And they occur all over the United States, from freshwater to saltwater systems. The specific impacts of harmful algal blooms will vary based on the species, the location, the time of year, and the proximity to these important resources. The main ecological impacts I already talked about. It can cause low dissolved oxygen regions. It can cause food web disruption, and it can block sunlight for submerged aquatic vegetation that is below the phytoplankton. The economic impacts include loss of recreational revenue through beach and lake closures. For instance, officials in 2019 closed all of the Mississippi beaches due to a blue-green harmful algal bloom. As you can imagine, this has terrible effects on tourism for the region. It can cause increased water treatment costs. For instance, in 2014, the Toledo water crisis occurred, where Toledo lost its use of water for a while because of a bloom that was happening in Lake Erie. And these blooms happen annually, and the water treatment facilities have to pay a lot of money to treat this water, so it's to standards for us to drink it. And then it can also have impacts on fishing, crabbing, and shellfish bed closures, because shellfish filter all of the water, filter the water very well. They're filter feeders. So they take up all of that phytoplankton. And in doing so, they're taking up its toxins as well. So we don't want to eat any shellfish that has a lot of the toxin in it. So we close the shellfish beds. Which leads me to health impacts. So we can have direct exposure to the toxin through swimming and through the air. We could have contaminated shellfish, contaminated drinking water, and you have to remember dogs as well and animals. They'll swim in the water, and when they swim in the water, they'll lick their fur, they'll chew the water as they're chewing a stick, and a lot of dog deaths have occurred from harmful algal blooms. Which leads to the second act of our talk today. What harmful algal blooms occur in Maryland, aka the villains? And I'll hand it over to Al.
Right, so as you see in your slide, Loki is green, representing our cyanobacteria. Guys, across the country, our waters are becoming greener and greener. And the bad thing about all of this is basically they produce really nasty toxins that have long and short-term effects, everything from inducing cancer and so forth. Here in the Chesapeake Bay, we have problems with green water on the eastern shore because of agricultural input, and also just because of general increase in temperatures that are occurring around the world, basically. These organisms are really interesting. They're single cell, they're bacteria that just happen to do photosynthesis, and they have the ability to actually, they have a gas vesicle that they can inflate, so they can actually, even though they're not modal, that is, they don't have any way of moving themselves, they can come to the surface, and you see this as scums. So my first advice to you guys, if you've seen green in the water, especially if it's streaky like this, don't let your dogs or yourself go into it in the process. In the eastern shore and throughout, the major player that we have is an organism called microcystis, and it makes these very potent microcystin toxins whose function ecologically we still really don't have a good handle on. But in terms of water treatment, and this is the major problem that has occurred in lake areas, Taylor's already talked about. So they produce hepatotoxins, neurotoxins, or skin toxins in the process. And we have a really good ability to now assay these both in the water systems as well as from the treatment of water. But if you see duckweed on the surface, that is not necessarily bad. That's actually probably a good indication that the water is reasonably stable. Next slide. So for me, I actually got into this field because of an event for those that are around called Physteria back in the late 90s, basically. We had a fish kill on the eastern shore in the Pocomoke, basically, and some watermen were affected. And it was supposedly due to an organism called Physteria piscicida. But in fact, 15 years of study later, basically, we found it's due to this other organism, which has its name Beneficum, which means toxin producer, which was originally described in the 50s. And we've been able to show this is a really interesting plant that eats other plants, and it uses the toxin to capture those plants. In the meanwhile, when it blooms, basically, it causes fish kills because the toxin hits the gills of menhaden in the process. For those who've been around long enough, we have these brown tides due to Procentrum minimum. You see over on the right, basically, in our harbor, basically, a very dense brown mahogany tide is referred to. And for those of you who have been keeping up, in the south part of the bay since 2007, we have a introduced warm water species called Alexandria monolotum that not only produces a toxin that currently we don't have a clear effect on human effects, but it clearly has effect on mollusks and so forth. But it also bioluminescence, which is a really interesting, you see here in the next slide, where the VIMS workers are actually able to spell the word out HAB by simply essentially scraping on the sand basically in the process. So you don't have to go to Puerto Rico to see these. If you go in June in the York River, you'll basically see big bioluminescence. So this is this members of the Dinoflagellates, which kind of defines what they are. They have two little whip-like flagella one around their belly and one coming out their butt. Next slide. So this is probably the worst one. Fortunately, it's never gotten to toxin levels. On the West Coast, there are these organisms called Pseudonychia that produces this really bad toxin called domoic acid. And this is what's responsible for problems with seals in terms of literally destroying half of their brain, closing the crab hatchery, crab fishery, basically, which has a big effect around this time of the year. This doesn't have effect on humans, especially low levels, and it's been shown. We're still not at a level of having to worry about toxicity. And for those who have read the storylines of the birds, it's thought that actually birds that eat anchovies gave rise to this idea of the behavior of swimming, flying at people basically in the process. So currently it's not an issue on the East Coast, but it is a big issue on the West Coast, and it's becoming an issue in the northern latitudes around Woods Hole and so forth. Next slide. Is it you? Yes. So what causes harmful algal blooms? I'll now leave it to Taylor.
Yeah, so like in the latest Avengers movie, the villain Thanos becomes all-powerful when he collects these 6 Infinity Stones. Like Thanos, harmful algae are our villain, and they become all-powerful when they have optimal light, temperature, salinity, water flow, nutrients, and minimal competition and predation in the water. This is where we will get into the discussion that harmful algal blooms are naturally occurring, but humans are doing some things that are increasing their occurrence and helping them outcompete the good phytoplankton. So light availability. Like all plants, algae have chlorophyll, which is a pigment used to capture light for photosynthesis. They need light to function, and phytoplankton actually produce 80% of the world's oxygen. They also all grow on an ideal water temperature. Many harmful algal bloom species like warm summer seasons with long light days, which is why we tend to see more about harmful algal blooms in the news during the summer. Like atmospheric temperatures, water temperatures are warming due to human greenhouse gas emissions. And this can actually benefit some harmful algal bloom species because at a higher temperature, they have greater growth rates and can dominate over regular phytoplankton. In addition, it can lead to longer bloom seasons, and they can spread to new areas where it was too cold previously, like in northern Alaska, we're seeing a lot more harmful algal blooms. Each species of phytoplankton and harmful algae also have their ideal salinity, so their salt content. So we already talked about how there are freshwater harmful algal species and saltwater, and they all have their specific salinity that they like. They also all have a water flow rate that they like as well. If nutrients are limited, which they need nutrients, we'll get to that on the next slide, water movement can bring in more nutrients through upwelling or through rain events. Some species, including a lot of toxic species, can vertically migrate, meaning they have a competitive advantage over others in calm waters because they can go towards the surface to get light, but then can move down to get ideal nutrient content, which other phytoplankton cannot. So toxic algae tend to like calm waters. There are also direct human impacts to water movements that we're doing. So we're introducing new species to new areas through ballast water and shellfish farming. As we transport the shellfish, as we transport the boats, we're also transporting the phytoplankton and the algae that come with it. and also through hydrological modifications like dams, where when we put up, this is a picture of the Conowingo Dam, and this water in front of the dam is calm and nutrient rich, and the algae are just loving it. Which brings me to nutrients. So humans are adding excess nutrients into the water through point source, so wastewater treatment plants and factories, and non-point sources, so agriculture, urban road runoff, chicken farms. And we're primarily talking about nitrogen and phosphorus. And this just fuels the bloom for a long period of time. Once the nutrient runs out, they stop growing. So as long as they have nutrients, unless something else becomes not optimal, they can keep growing. And so the best way to reduce harmful algal blooms are to reduce these nutrients. They also need minimal competition and predation. So competition like macroalgae and other phytoplankton, they can compete for nutrients and light and other sources. So if they're not there, then harmful algae don't have issues. They can also release what I'm calling fighting chemicals. So they can release certain compounds that actually inhibit the growth of harmful algae or the other organisms. Predators such as zooplankton and shellfish can help keep harmful algal species at a controlled concentration, but some actually selectively feed, so they can remove the competition and remove these other good phytoplankton and let the harmful algal blooms just go crazy. An example of this is actually the invasive zebra mussels that were introduced to the Great Lakes that seem to not like microcystis, the toxic cyanobacteria that other shellfish are willing to eat. Now we will discuss what can be done to combat harmful algal blooms. In order to protect us from harmful algal blooms, scientists are using models to forecast when and where a harmful algal bloom will occur and for how long. This can reduce human and resource exposure, such as giving water treatment and shellfish facilities warning to stop flow into their facilities. Prediction and forecast can also show scientists where they need to be monitoring and when. And there are some innovative ways that scientists and managers have started monitoring harmful algal blooms. They've started using satellites, they use buoys, they use sensors, and the good old-fashioned grab sample where they just grab the water, look at it, and identify the phytoplankton. Monitoring for harmful algal blooms can lead to early warning and closing of these beaches and shellfish beds to ensure public safety. And then there's what I'm going to be talking about mostly today, control mechanisms. So these are generally limited to ponds and small lakes because in large-scale systems, it's difficult to balance this reducing harmful algal blooms with its unintended side effects that the treatment options might cause. So there are physical, chemical, and biological methods to controlling HABs. Most of these methods look at controlling one of the optimal conditions that I talked about, whether that's light, water movement, competition, or temperature, or physically removing the cell. So I'm just going to mention a couple of the control mechanisms. There's more out there, and they all have their own benefits and drawbacks. So there's aeration, where you're adding literally air, adding bubbles into the water, and this causes water movement, brings the temperature up, so it's not necessarily an ideal temperature, and changes the light. Mixing does the same thing, just physically mixing the water. As you can imagine, you can't really mix a whole coast. At most, you can mix a small lake. Then there's dredging. Dredging can be used to reduce the legacy nutrients, so nutrients that are in the sediment that can be released and help keep a algal bloom going. But the drawback of dredging is it can also introduce a lot of sediment into the water column and contaminants that were in the sediment that you're then just releasing out. So pros and cons of dredging. And then a very popular method, and one of the few methods that's been used for both small and large systems, are modified clay, which the clay can bind to nutrients, it can bind to cells, and then ostensibly get rid of the bloom. The chemical treatments right now, the most popular are hydrogen peroxide or copper sulfate. The problem with these are it works really well, but it works too well. It attacks non-targeted organisms, and so it just pretty much knocks out all the phytoplankton. And then I'll leave Al to talk about the barley straw on the next slide. Then there are biological mechanisms, so adding predators and adding macroalgae and submerged aquatic vegetation, and then maybe it reduces the nutrients and reduces the harmful algae. So there's not a whole lot of drawbacks, except it won't necessarily work 100% of the time. And then there are bacteria and viruses that release these fighting chemicals. Scientists are studying certain bacteria that seem to kill just a specific type of algae. And they're also looking at separating out the compounds that are killing that the bacteria are producing and just exposing the algal bloom to those compounds. So there are many techniques out there, and they all have their pros and cons. But in order for there to be an ideal technique, it must be effective, it must not cause any side effects, and it must be cost less. It must be low cost and be okay for society. And there's just not a lot of current techniques that meet all of these criteria. And all HAB species are not equal. One technique will move the water, but if you have a toxic algae species that likes water moving, then it's not necessarily going to work. So the best technique varies based on the species and the water system. And like Iron Man attempting to fly for the first time, scientists and managers have studied and gone through a lot of crash landings. And so if you do have a harmful algal bloom in a nearby lake or a pond, work with scientists and managers, and they can help guide you on what technique you should use. Now I will pass it back to Dr. Place to talk about what our lab is working on to combat harmful algal blooms. So
as I said, in the late 90s, basically, our lab focus was primarily on detecting what algae are toxic, what they make, and how we can assay for that. We moved in 2011 with NOAA funding, basically, to begin to what kind of treatment options could we do. And basically, we were luckily enough to find a private lake owned by the Girl Scouts of Delaware and Maryland called Camp Todd. And you see in the middle, when we arrived there on 2011, what was happening. Completely green pea soup, basically. So bad that, in fact, the Girl Scouts were on the beach in front of this lake you see, learning the J stroke, and then had to pick their canoes up and go to another lake to actually do it, because the toxin levels in this lake at that time basically were sufficiently high to actually cause issues. Mother Nature helped us in treatment options. We had a 3-year study on this lake, basically, and for 3 years we had hurricanes Irene, Tropical Storm Sandy, and Tropical Storm Andrea that were coming through. And in order for this lake to not wash out the road, the owner basically had to open the dam and allow the lake to be basically draining. And we actually asked him, well, let's try something. Let's actually leave the lake drain because it was closed over the winter and basically into the spring. And so during that draining, basically 85% volume, or 80% of that volume was washed out. And this is like the perfect way to kind of knock down a harmful algal bloom is dilute it out and force it to grow up again next year. So we were able to do that for 3 years. And the other thing we did was actually based on British findings that barley bales, barley, specifically barley, not hay, when it rotted, basically, it produced a material that it basically inhibited this. And this was all actually, we had solid data nearby. There was a small farm pond where the farmer had been putting leaven bales out, and he didn't, at the same time, he did not have a microcystis bloom in the process. The thing we kind of began to find is this idea that this breakdown material are these molecules called tannins or flavonoids in the process. And actually, this is where Taylor got involved. Taylor realized, well, what happens when you make beer? You use barley. And what happens with all this barley distillate solids at the end in the process? So now with new funding, basically, we're beginning to look at what is the nature of the materials in this distillate solids? And can we now use that material as a way of inhibiting the growth like we did with the barley bales, which required 30 people to carry these 40-pound things while the lake was drained to put in the tributaries. And it only is effective for a short period of time to that top part that remains dry in the process. Next slide.
Yeah, so I'll take over and talk about the Brewer spent grain idea.
Yes.
So, as he mentioned, there are some drawbacks to using barley straw. They're heavy, and most of all, you have to put them out before the bloom even occurs. It might not even occur that year. You have no idea, but you have to put these out because it takes months before it starts degrading and releasing these compounds that are inhibitory to the algae. So I thought when you think barley, you think beer. And so could a byproduct of breweries be used as a better method than barley straw? So to give you a brief overview of the brewing process, breweries take a variety of grains, but primarily barley, the barley husk, which you can see right here. This is the brewer spent grain. And they grind it up, they heat it, they mix it in with hot water. They let all of those delicious sugars that you love about beer come out, and that moves on to the next process. And then they're left with all of this wasted husk. So this is the brewer spent grain, and there's 4.5 million tons per year in the US made. And there's not a whole lot of uses for them. Most breweries actually give them, donate it for animal feed to give to cows and chickens and pigs. But a lot of it ends up in landfills because there's just not, people are actively looking for alternative uses of brewer spent grain. Sorry about that. So our lab took brewer spent grain, mixed it with water, filtered it, and then exposed it to different algae. And it actually, I also forgot to mention, the barley husks actually have 5 times the concentration of these flavonoids, these phenolic acids that we think are the inhibitory material compared to barley straw. So we had reason to think that it would even work better than barley. So we took this water and this extracted Brewer spent grain water, and we exposed it to a toxic dinoflagellate, a non-toxic green algae, and a toxic cyanobacteria freshwater algae. And you can see over day, we counted the cells per milliliter over time, and we had a control where just plain water was added. Our barley in purple, our spent grain in blue, and sargassum, which is a type of brown macroalgae, in red. And what we found was for the toxic dinoflagellate, spent grain just immediately killed them. And same with the toxic cyanobacteria. But with the non-toxic green algae, it actually increased growth. So we'll redo the non-toxic green algae, but this is promising that it does not seem to harm non-toxic species. However, when we sent off the water extract for nutrient analysis, we found that there was very high levels of ammonium. So you don't want to add the Brewer spent grain straight to the water. We really want to extract out these compounds first. We ran another experiment where we exposed a bloom that was happening in Lake Williston of a potentially toxic cyanobacteria. And it was pretty much straight cylindrospermopsis, as you can see in the control picture. And we either dosed it with 0.1% of this spent grain extract or 1%. And over time, the 0.1% actually ended up having a higher cell per mil of cyanobacteria. The cyanobacteria you can see in the red, orange, and yellows. And then the non-toxic species are in the green, purple, and blue. So 0.1% increased it and kept the bloom going, probably because of this extra ammonium and these extra nutrients. And the 1% immediately pretty much killed it off. By day 4, it was pretty much nothing. And the non-toxic species were able to then bloom, as you can see in the 1% spent grain picture. So the conclusion of this study was that dose matters. We can't just arbitrarily add Brewer spent grain, because if you add too little, they'll benefit from the nutrients, but there's not enough inhibitory material to actually impact the bloom negatively. And then I'll pass it back to Dr. Place to talk about our flavonoid experiments.
So given that we believe the active component in spent and also the barley rotting are these compounds called flavonoids. These are molecules right now that are having big play in human nutrition, both positive and negative effects. So they're commercially available as food additions. And the take home from this is that these different structures that we show here, some are very, very effective on some species and some are not very effective. So with recent funding from Moat, we're now looking at the effect that these molecules could have potentially on the classic red tide blooms that exist in Florida for those that are down there and have to deal with that as a respiratory material. So we'll tell you more about this in the future, but the bottom line is this is a material that is commercially available, relatively inexpensive, and has both positive and sometimes negative effects. Next slide. So it should be clear to everybody, these are all stopgap treatment options. The ultimate goal is, can we reduce the nutrients that are driving this? We, early on with our NOAA funding in Lake Williston, we investigated one of the treatment options. And the general belief, although I believe it's both nitrogen and phosphorus, is that if we can lower the phosphate in these tributaries coming from either wastewater treatment options or agricultural fields and so forth, we should be able to suppress the growth of at least the cyanobacteria. So a reagent that we use is a clay that the Australians have patented, basically, where they replace the calcium ions with lanthanum. And it turns out this material binds very, very tightly, phosphate, both in solution as well as sometimes in soils in the process. The problem with this is that if we were to treat the lake Williston, we had an estimate on this, and we actually evaluated it. It does work. It would have cost about $150,000 to treat that lake per year because you have to continually do this. And the other interesting thing we found is basically, yes, it bound phosphate, but then it made the sediment more anaerobic so that we had a big increase in ammonia coming out of that environment. So the treatment option was not a valued one. What's really interesting recently is we moved to a natural material. If you've ever down in the Bahamas, you notice how white their beaches are and everything else. Well, there's this natural material that gets precipitated by biological systems, actually by cyanobacteria, with the right amount of CO2 in the environment, as small crystals of calcium carbonate, as aragonite. And you see this material right here. Turns out this material also binds phosphate, but in a much less tense way. And in fact, you can put this on an agricultural field, and in principle, and with some lab experiments, it actually lowers the leaching of phosphate from the soil, and hopefully would make that available to the plants. So this is a current big finding we're working on right now, is using this natural, organic, not man-made material that's available in millions of metric tons because it's annually being precipitated. And it's being brought up by a company basically for agricultural and wastewater treatment options. Next slide. So in summary, Taylor will say.
So in Act 1, we found out that harmful algal blooms are bad and they cause ecological, economic, and health impacts. In Act 2, we found out that harmful algal blooms occur all over the United States, including Maryland. In Act 3, we found out harmful algal blooms naturally occur, but human impacts are causing them to increase in frequency and duration and spread to new locations. And in Act 4, we found out that scientists and managers are working on ways to predict and combat harmful algal blooms. But it's hard to monitor and protect all areas and not to harm ecosystems in the process. And our overall conclusion is that video games can be very educational. You should definitely go and learn how to fight harmful algal blooms from Spider-Man. So we're hoping that you can answer the question, when did you become an expert on harmful algal blooms, as tonight?
Yes, tonight.
And don't forget, we can all be superheroes for the environment. Who you thought was going to beat Thanos in the last movie and who's really going to do it.